take a look at the process of transcription. This is where a new strand of RNA is going to be synthesized using DNA as a template. As stated previously, there are many similarities to the process of transcription with respect to DNA replication, such that both of these strands of DNA need to be pried apart, specifically meaning that the hydrogen bonds between the two strands must be broken. In this case, however, we will be forming a transcription bubble, rather than a replication bubble in the DNA replication process. Note that in this process, we are going to be dealing with the enzyme RNA polymerase. This is the important enzyme that will make the new strand of RNA, and it will do so in a complementary manner to the DNA template strand. Keep in mind, as stated previously, RNA polymerase acts very similar to DNA polymerase, such that it can only read a strand in 3 to 5, and can only produce a new strand of RNA in 5 to 3. RNA polymerase will slide along the template strand in a 3 to 5 direction, creating its new RNA strand. Note that this overall process is similar with respect to prokaryotic and eukaryotic cells. However, an important difference in eukaryotic cells is the way that RNA is produced. Eukaryotic cells have 3 different types of RNA polymerase. Each one will be responsible for making a different type of RNA. Remember, we have mRNA, rRNA, as well as tRNA. So in eukaryotic cells, we will have specific RNA polymerases that are involved in making these specific RNA molecules. Please note, a common confusion for students is in thinking that these 3 versions of RNA are not present in prokaryotic cells. However, prokaryotic cells still need to undergo transcription and translation to produce proteins. As a result, they must have mRNA, rRNA, as well as tRNA present. The difference lies in how these RNA molecules are produced. They're going to be produced by 3 different RNA polymerases in eukaryotic cells. This we do not see in prokaryotic cells. Prokaryotic cells, however, only have 1 type of RNA polymerase. However, they still possess all 3 types of RNA. The process of transcription can be divided up once again into 3 different phases: initiation, elongation, and termination. This diagram here depicts the overall process of transcription with a closer examination of the RNA formation. Note that we have the 2 different strands of DNA, one running in 3 to 5 and one running from 5 to 3. In this case here, the direction of transcription is moving from left to right. As a result, the bottom strand is going to be considered the template strand. RNA polymerase will be able to bind to the DNA. It will be able to take those strands apart, dissolve the hydrogen bonds to be able to access the DNA template. It will then begin adding the complementary base pair sequences using RNA nucleotides as a base. Remember, the RNA nucleotides would have ribose as a base sugar. We designate the ribose-based nucleotides as being in red in the diagram, whereas the DNA-based nucleotides are designated in blue. We can see that the newly made RNA transcript is being produced in the 5 to 3' direction. We can also see the complementary way that the bases are added. When we have a T in our DNA template, we would be adding an A to our RNA strand. If we have an A in our DNA template, in this case, we would be adding a uracil or U to the complementary RNA strand. The process of transcription, like many cellular processes, is highly regulated. Genes are only going to be considered to be in the on position when they are actively being transcribed. There are many different sequences on the DNA molecule that assist in regulating the process of transcription. One of those important regions is the promoter region. This is the sequence on a gene where RNA polymerase will bind. This sequence here designates the start of the transcription process or initiation. The promoter region tells RNA polymerase which strand to use as a template and in which direction in which to move. In prokaryotic cells, RNA polymerase can find the promoter region directly. This is because prokaryotic cells have a very simplified genome. Most of the genome is going to contain important DNA sequences. In eukaryotic cells, however, we have very complicated DNA that can be wrapped around in histone proteins, which this can make it very difficult for RNA polymerase to find the promoter region. As a result, in eukaryotic cells, there will be the need for transcription factors. Transcription factors are proteins that will help recognize where the promoter region is. They will function in being able to recruit RNA polymerase to the promoter region. Thus, I consider them to be the neon signs of transcription. They help to highlight the promoter region and tell RNA polymerase exactly where they need to bind to be able to transcribe the important genes of interest. The promoter acts as an on switch because transcription begins here, but it also can act like a dial to fine-tune control. Remember, transcription factors can act to turn on and off genes. However, they can also act to upregulate or downregulate the process of transcription. Within the promoter region, you will have typically what is termed the Tata box. This is a specific DNA sequence that is highly conserved among many different species. This DNA sequence can in fact be found in most promoter regions. There is also the DNA sequence that designates where transcription needs to stop. We refer to this as a terminator region. This diagram here gives an overview of this process. Note that we have a promoter region which lays upstream or slightly before our genes of interest, which are going to be transcribed. Note that in the top of this diagram we have the TATA box denoted in the promoter region. Also note that the sequence TATA, T-A-T-A-A-A-A, can be found in the non-template or coding strand. This indicates that the actual sequence in the DNA template strand for the TATA box would be the complementary sequence, ATATTTT. Together, those two sequences designate the TATA box, which is that highly conserved region commonly present in most promoter sequences. Also note that we have a start point to the transcription process located further along the gene, slightly after the TATA box, but before the actual gene of interest. This is very typical for a eukaryotic promoter region. Because we are dealing with eukaryotic DNA, transcription factors will be involved into binding and recognizing the promoter region, specifically being able to recognize the TATA box. These transcription factors will assist in regulating the transcription process. Many times in eukaryotic cells, you will have several different transcription factors that help to highlight the promoter region. Once the promoter region has been highlighted, RNA polymerase can now recognize the area. They will be recruited to the area where the transcription factors are bound. They will be able to bind to the promoter region, and thus they will be able to then slide along the DNA template to create the new RNA transcript. There are also many important differences in the process of transcription and specifically dealing with how the RNA is processed or how the RNA is used in the cell. In prokaryotic cells, remember the genome is very simple. This means that as soon as the RNA is constructed, it can be translated immediately. There is no need to process out any information. Whatever is in that RNA molecule is going to be important for translating a protein. This means that the process of transcription and translation can happen simultaneously at the same time within the cell. Also note that the location of this all occurs in the cytoplasm because there is no nucleus in prokaryotic cells. In eukaryotic cells, though, we have a different story. The eukaryotic genome, as stated earlier, is much more complex than a prokaryotic genome. This means that the RNA that results from the transcription process needs to be processed in order to become a mature mRNA molecule. Part of that processing involves three important steps. One of those steps is to remove the introns. When we have a primary RNA transcript, it will consist of both introns, which is intervening information, as well as exons, which is the important expressed information. In order to be processed, the introns need to be removed and the exons need to be glued together. This is to ensure that the proper information is translated into a protein. Two other important modifications are involved in protecting the RNA transcript from degradation. These two modifications include a modified guanine being added to the 5' end of the RNA transcript. We call this the 5' cap. The other modification is to create a poly A tail at the 3' end of the transcript. This simply refers to the fact that a sequence of adenines are going to be added to the RNA transcript on the 3' end. These will help, once again, to protect the RNA from degradation. This will also help to facilitate its export from the nucleus. Remember, the process of transcription occurs in the nucleus. However, translation occurs in the cytosol. So once an mRNA transcript is processed, meaning the introns are removed, and it is protected, meaning that the 5' cap and 3' poly A tail is inserted onto the transcript, it can then leave the nucleus to be used for the translation process. We're now going to take a closer look at the process of mRNA splicing. Splicing refers to the fact that the RNA transcript that is originally produced from the DNA molecule needs to be processed. The introns, or intervening information, needs to be removed, and the exons need to be kept. Exons refer to the specific regions of DNA and mRNA that code for the protein, whereas the introns refer to the larger portions of DNA and RNA that do not code for protein. Genes can have many different alternating exons and introns within their overall sequence, but the final mature mRNA that eventually will be translated has all the introns removed. This is done using an important complex called the spliceosome. The spliceosome is an important complex that has many small RNA molecules attached within it. These small RNA molecules are important in recognizing where the introns exist in the pre-RNA transcript. We can also call this transcript a primary RNA transcript. Once the introns have been identified, spliceosome components can come in and excise out the intron. They will also help to glue the remaining exon sequences together.